Hello Year 9, this is part 1 of 3 parts revising the topic that we've just finished learning, B4, bioenergetics. Okay, now within the sheets that I sent you in the flashcards, you will see 3 sheets. Okay, so this is the first one and this is what we're going to fill in today using this video. Okay, so I'm going to talk through the content that covers everything on this sheet. If you can't print this, then you will have to just create from scratch your own. It will take a little bit longer, but it can be done. My class, I gave you these 3 sheets just before we left school, so hopefully you can use those. And we're going to be looking at the limiting factors of photosynthesis. We're going to be looking at the uses of glucose in plants. We're going to talk about the required practical, how light affects the rate of photosynthesis, the photosynthesis equation, structure of a leaf. So that's what's going to come up in today's first of 3 revision PowerPoints. You've got the flashcards. By all means, if you've got the ability to print the flashcards, then do. You can print those so you have 2 slides on a page, and that prints as quite a nice set of flashcards. Okay, so let's have a look at what we're covering for this particular sheet. So we started off at the beginning of the topic talking about structure of the leaf. We said that the leaf is thin because if it was really thick, then the light wouldn't go through to the chloroplast and wouldn't be absorbed by the chlorophyll, and photosynthesis would be less effective. They're broad, so that means really kind of wide and flat. They've got this network of veins which comprise of the xylem and the phloem. Here we can see the xylem carrying the water and the minerals through the process of transpiration. That happens in one direction from the roots to the leaf, where water evaporates out of those stomata, and we call that transpiration, as I just said. We did cover that in B1 as well. I like to keep talking about it to keep that fresh in our minds. The other vessel in the veins is the phloem, that's this one here, and that transports the sugar to all locations. Transporting to different locations, the process is called translocation, and the sugar is made in the leaf, but all of those cells within the plant, from the roots to the stem to the flower, every cell there is going to be or pretty much every cell is going to be going through respiration, therefore it needs that sugar. It needs the sugar for the construction of cellulose, for amino acids, all those things. We'll talk about those in greater depth shortly. We've got the chlorophyll, that's what makes the leaf green. Found in the chloroplast, absorbs the light for photosynthesis. Flat, we've said already. They don't overlap. You don't see 2 leaves on top of each other like this because the one underneath wouldn't get any light. We've got this waxy cuticle on the surface of the leaf. That makes it waterproof, stops any water from evaporating from the top of the leaf. We don't find the stomata in the top of the leaf, only on the underside of the leaf. We've got the palisade cells. That's the one that people forget the name of. It's the palisade palisade cells, just this layer here, which is our typical photosynthesizing cells. Okay, so the palisade cells have the highest density, highest number of chloroplasts for the photosynthesis to occur, and that's really important. They're near the top. We've got this upper epidermis here, lower epidermis here. They are just protection cells. Okay, they are just helping to protect the leaf. And then underneath that, we've got the palisade cells. Below that, we've got these odd-shaped cells called the spongy mesophyll. Some chloroplasts still found in that area, and so therefore some photosynthesis occurs there. And then down in this lower epidermis here, we've got the guard cells, okay, which can open and close depending on how dry or wet it is, and their primary function is gas exchange through the stomata. Okay, one is called the stoma, many is called stomata, protected by those guard cells. At the same rate, at the same time, should I say, water can escape through those holes. But that's not the purpose of stomata. The purpose of stomata is gas exchange. It just so happens that if you have lots of holes in a leaf, water will evaporate through that. We talked about to create that word and symbol equation, we talked about the fact that carbon dioxide goes into the leaves, water goes into the roots. So those are our reactants, the things that are going to react during photosynthesis. It needs the presence of chlorophyll, the presence of light. And we talked about those items being kind of like the oven, okay? You need the oven to bake the cake, but it's not a reactant, okay? So the carbon dioxide, the water, they are our reactants. The light and the chlorophyll need to be there for the reaction to occur. And then produced from that is the oxygen, which is released by diffusion, and the glucose, which is used by the plant. Okay, so if in that we think, oh, I can't remember the word equation, the symbol equation, you can kind of work through in this logical process. Sorry about that, I was chatting away and the phone randomly sprang out of the tripod and I caught it in my hand. So we've got a bit of a break there in transmission, but I'm sure we'll be fine. So we said that light energy is absorbed by the chlorophyll. Because that light energy is needed for photosynthesis to occur, we refer to photosynthesis as an endothermic reaction. Okay, it might be that you want to pause now, write any notes that we've come across onto that revision sheet or your own revision sheet. If you would prefer to do it in a different way, not use the sheet I've given you, that's absolutely fine. If you'd like to make your own version of flashcards, absolutely fine. Okay, so the photosynthetic reaction, it's a chemical reaction, takes place in the chloroplasts where, and this is just kind of a recap really, carbon dioxide and water are converted into glucose and oxygen. And photosynthesis requires enzymes, okay? So we know that enzymes are important in the body. We learn more about that in B2. But photosynthesis also requires enzymes. And that links in to what we'll learn about temperature and how temperature will affect the rate of photosynthesis shortly. Chlorophyll is this pigment found in the chloroplast. You can see down here we've got our chloroplast. This is from probably an electron microscope, and these are all the little chloroplasts in that one cell there. We've also got a variegated leaf down here. We talked about the variegated leaf. It's the yellow parts or the white parts, no chlorophyll. So no photosynthesis can occur there, no glucose found there, no starch stored there. They're kind of redundant parts of the leaf. So we had our word equation, carbon dioxide plus water, the light and chlorophyll being like the oven, they need to be there for the reaction to work, and glucose and oxygen being the products. And here we have our symbol equation. Okay, so glucose is C6H12O6. It's kind of like a 6 sandwich. Okay, and then you put the two bits of bread together and you get the 12 in the middle. And in order to balance it, rather than wasting loads of time trying to balance it, we just put a 6 in front of all of the small molecules. So we've got a 6 here, a 6 here, and a 6 here. Okay, so like I said, pause where you need to, add any notes that you need to your sheets or your own independent work. So we spent a lot of time using the Focus software, which looked a bit like this, to investigate rate of photosynthesis. And to measure rate of photosynthesis, we were actually measuring the amount of oxygen produced in a given time. So we went for 10 minutes and we looked at how much oxygen had been produced in that time. We used pondweed because that lives underwater and we could see that oxygen coming off of the pondweed. You could alternatively, and I've seen this in exam questions, you could count the bubbles of oxygen produced in say 30 seconds or a minute, okay? And that would be slightly different from collecting the oxygen in a syringe, but it would do exactly the same job. Now we're looking at all of these different graphs, okay? And light intensity, as light intensity increases, the rate of photosynthesis increases to this point. Now that means that more oxygen was being produced as the light intensity increased. And then we get to this kind of best point, which we call the optimum. When we increase the light intensity beyond that point, imagine like a dimmer switch, or you put physically more lights onto the plant, you're spending more money on the electricity for the lights, but actually it's having no impact on rate of photosynthesis. So to put ourselves in the position of the farmer, that's really important. We don't want to be spending extra money and yet getting no extra profit as a result. Okay, same things happens with carbon dioxide. So if this was a greenhouse scenario, we could pump more carbon dioxide in and it would increase the rate of photosynthesis up to that optimum point. It's going to cost more money to get the carbon dioxide increased any further, and yet it's having no profitable impact because there's no extra photosynthesis, therefore it's not improving growth anymore. Now, as soon as one factor hits the optimum, something else becomes a limiting factor. Okay, so once light intensity is at the optimum, it's no longer a limiting factor, something else has become a limiting factor. Now, temperature. This relates to those enzymes, okay, and we haven't learnt about denaturing of enzymes yet, but we will learn it in B2, which is at the start of year 10. So think kind of back into your chemistry mindset. As particles gain energy, they move around more, therefore there's more collisions. So that's why as temperature increases, the rate of photosynthesis increases, because the water and the carbon dioxide are more likely to collide, react and produce those products. When we've got beyond the optimum over here, this is like 40 degrees and beyond, enzymes start to denature. They basically change shape. They're not able to do their job properly. And I'm not going to talk lots about denaturing because we haven't done the topic where we learnt what denaturing is. But that is why get them too hot. Obviously all the water would evaporate out of them as well. They'd wilt. But it's the enzymes that's the important thing. And chlorophyll, the more ovens you've got, the more photosynthesis can happen. Okay, so the more chlorophyll there is in the leaf, the faster and the more photosynthesis that will occur. Okay, so we've got some boxes where we can either write those notes, we can write our own notes, we can think about the things I've talked about and add those to our notes. Okay, if you need to pause, do so now. Now, this bit is for higher only, okay, so this will help Sarah's group. My group, we haven't learnt this, but basically what we're looking at here, it's a little bit difficult to get your head around. We'll just focus on the left-hand side for now. It's analysing graph with lots of limiting factors. And if we just look at this line at the bottom here, if we start off with this low concentration of carbon dioxide at a low temperature, we find that light intensity stops being a limiting factor and the optimum is about here. Okay. However, if we just thought that and thought, okay, well, I know what my optimum for light intensity is, I'll go ahead and increase carbon dioxide now. Okay, you increase carbon dioxide and you can see that light, actually when you retest, it becomes an optimum at a much higher light intensity. So what we're really learning from this is that we have to keep repeating our tests. We can't just say, okay, well, we've done light intensity, we know the optimum, right, now we're going to investigate something else, but we know that our light was this. We have to say, okay, well, now we'll find out what's the impact of carbon dioxide, what's the impact of temperature, and then really we should go back and when we've got our optimums, retest light. Has light changed? And yes, it has. The optimum's gone from down here all the way to over here, which is, you know, a good halfway across the light intensity scale at those new conditions. Okay, and that's what's really important to understand. We've then got this thing called the inverse square law, which looks a lot more complicated than it really is. Basically what we're saying here is if I move the lamp 4 times the distance away, if I do 4 squared, that's 16, it will be 16 times less bright. Okay, so I've moved the lamp 4 times the distance away from where it was originally. 4 times 4 is 16, so it'll be, the light intensity will be 16 times less bright. Okay, and if I move it 6 times the distance away, 6 squared, 36, it will be 36 times less bright. Okay, so the grid there is just really to help us with our squaring of numbers. Okay, so this page here is higher only, so some of you won't necessarily need to look at that. You can if you want to, even if we've not done it in class, you're more than welcome to write notes on it. If you need to pause, pause now. Okay, Uses of glucose from photosynthesis. Now, I told you this way of remembering it, which is scarf. The exam board do not know what scarf means, so you have to spell it out. It's just my little trick for remembering the 5 uses of glucose from photosynthesis. So the plants are doing all this photosynthesis, they are making this glucose, and scarf helps us to remember how they use that glucose. So some of it is stored as starch. Okay, can be used when it's dark, when there's no photosynthesis, that midnight feast that we talked about. Some of it is used to produce cellulose, which is used to strengthen the cell wall. Some of it is used in the production of amino acids, and amino acids then build up into proteins, and proteins are used for growth and repair. Some of it is used in respiration. All living things respire, so some of it will then go through the reverse of photosynthesis when they respire. And some of it is stored in fats, as fats and oils. Things like our sunflower seeds and our olives, we compress those and get the oils out of them. Okay, so scarf is our little trick to remember the uses of glucose from plants, and it's for plants. It's an exam board favourite. I don't know why, but they love it. So it might be like a 4 marker, and it says some of the glucose that the plant makes in photosynthesis is used to make cellulose. Describe 4 other ways that the plant uses the glucose. And there's your answer. Okay, let me just quickly flick. Yes, right, so that is the end of part 1. You're obviously going to spend a bit more time on that now, creating your revision materials. So that's part 1 done of 3. And we will come back next lesson to do the next sheet, the next part where we're looking at storage of starch and that difference between the kind of relative amount of respiration and photosynthesis in the plant. Okay, enjoy the day. The sun is, oh, I can't even speak. Enjoy the day. The sun is still shining. Get outside in the garden, and I hope you found that useful. Thank you.